Welcome to the second lecture in Micro 315. We're going to jump right into it today and start talking about the forms that bacterial cells take. Now, as I mentioned in the previous set of lectures, there is an enormous diversity of prokaryotes, and despite the commonalities among prokaryotes, the diversity that they house falls within the groups of form, so diversity of form, diversity of function, and phylogenetic diversity. So in the next several lectures, we're going to talk about each of those types of diversities and the commonalities and the differences between different types of bacteria. And we're going to start talking about form. Now, as I said in the previous lecture, it's very difficult to use things like shape to tell the difference between bacteria because bacteria generally take a handful of very common shapes. And the way that we refer to those shapes are as follows. The shapes can take the form of bacilli or rods, and you see two examples of that right here and right here. They can also take the form of cocci or spheres, and there are examples of that right here and right here. And cocci can either occur individually, so you have individual spear form cells. They can occur in pairs. That's often called diplococci, so if you have two cocci associated with each other. And they can also occur in chains. And you see this example of cyanobacteria where each cell occurs in a chain. Now, a couple of points about the spiral forms. So bacteria can take two different types of spiral forms. And even though they are both spiral, the mechanism by which the form obtained is different between the two forms. And I'm just going to briefly mention that. So first, let's talk about spirochetes. And you see an example of spirochetes right here. And in the spirochetes, let's draw a little line. In the spirochetes, the form is obtained through internal axial filaments and an outer sheath. So let me write that down. So those are the spirochete forms. And the other spiral form that bacteria take, Spirilia, are formed differently, and they're formed because of a wide, rigid spiral cell wall, similar to rod-shaped bacillus. So, I'll also write that down. Let me do it in a different color so that you can distinguish. So Spirilia, and that's a wide, rigid spiral cell. So in general, when you're talking about the shapes of bacteria, particularly say in the context of 315 lab, where you'll be looking at cells under the microscope, these are the forms that you will see them take. So let's talk a little bit about the bacterial cell itself. And here you see a picture of E. coli, and this is just a very basic picture. And in a typical bacterial cell, you have the cytoplasm of the cell, that's this purple stuff here, and it's surrounded by a cell envelope. And the cell envelope contains a membrane and a cell wall. And In bacterial cells do not contain a nucleus. So they don't contain a compartment surrounded by a membrane that houses the nucleus like eukaryotic cells do. Instead, there's a general region where the DNA is housed, which is called the nucleoid. And then you see stained here in these dark purple dots, that is the ribosome. So here I'm showing you the cross-section of a cell, and this particular cell is a gram-negative E. coli. And let me write that down for you. It's a gram-negative E. coli. Now, when we're talking about cells, bacterial cells specifically, there are three things in common. And the first thing that they have in common is a thick, complex outer envelope. If I can spell here. The next thing that bacterial cells generally have in common is a compact genome. That is, the genome, when you compare it with eukaryotic cells, is quite a bit smaller. There are fewer A's, T's, and G's, and there is very little non-coding sequence. So if you're used to studying eukaryotic organisms, there are commonly large introns in between the exons of genes. In bacterial cells, you don't have introns. And there are large intergenic regions, so that is regions of DNA that don't code for proteins in between protein-coding genes. And bacteria tend not to have large amounts of that type of sequence. And finally, what you see in bacterial cells is that functions are tightly coordinated. Which is particularly necessary because they don't have all of these organelles and nucleus and those types of things that compartmentalize different functions. Now, before we move on, I'm just going to talk about this diagram a little bit and talk about a handful of the things that are common to cells that I want to point out. Down here in the gray, you can see the DNA. And this down here is in the nucleoid. And then you have the cytoplasm. And notice that in the cytoplasm you have RNA polymerase. So that is, you're doing transcription from DNA into RNA. And in the blue you see the RNA. And here in the pink you see the ribosomes. So that is the cell machinery that translate the RNA into protein, and diagrammed here are the peptides that are emerging from the ribosomes. Now, up here you have the cell envelope, and in the cell envelope you have a lot of complicated things, and the exact components of the cell envelope will actually differ depending on whether the cell is gram-negative or gram-positive. But for the purposes of introduction, I'll just point out a couple of common features. And one of those is the internal membrane, so the bilipid membrane, and cell wall. So animal cells don't have cell walls, bacterial cells do. So the first thing that we're going to talk about in terms of pieces of the cell structure are cell membranes. And it is the cell membrane that defines the existence of a cell. And this should be a little bit of a review for you, but the cell membrane, like in eukaryotic organisms, is made up of a lipid bilayer. So you have a double layer of phospholipids, which you can see diagrammed right here in the picture. Here's the top layer, and here's the bottom layer. It separates the cytoplasm from the outside world, and proteins, which are shown right here as these lovely yellow blobs, are embedded in the membrane. And the proteins that are embedded in the membrane perform a lot of different functions. Some of those functions include anchoring the membrane to the envelope. So remember, the envelope isn't just the membrane. There are all sorts of different components like the cell wall. So it anchors the membrane to the envelope. There are receptors in there that sense the outside world, and the bacterial cell can adjust accordingly. And it can transport materials into and out of the cell. So let's just take a little bit of a closer look. This particular slide shows some of the same things, but in a little bit more detail. Now, this membrane, or the mechanical boundary, contains a bunch of proteins, like I said before, but here you see some proteins in a little bit more detail. And a couple of important things to note about the proteins is that it makes the membrane selectively permeable. So think about for a second what selectively permeable means. Anytime you have a bacterial cell, you need to be able to let certain compounds in, whether it be food, what have you, and you need to be able to transport waste out of the cell. That is, everybody poops. That is, every cell needs to be able to get the waste out of the cell. The bacterial membrane is also a site for the electron transport chain, and it's involved in lipid synthesis, and it contains receptor sensing proteins. So most of the proteins in a membrane are integral, so that is, these integral ones that span or are embedded in the membrane, and then the rest are peripheral, so that is, they sit on the edge of the membrane. So here we have a more detailed diagram of what the plasma membrane structure looks like. So as I said before, it's composed of a phospholipid bilayer. And these phospholipids are amphipathic. So that is, they have a polar and a nonpolar end. What you're seeing here is a diagram of the chemical structure of a phospholipid. So up here, here's the phospho part, and it has a head group. So this is the head group, and it's the polar and hydrophilic end, that is, it likes water. And down here you have glycerol attached to these long fatty acid chains. And these fatty acid chains are the pieces that form this inner part of the membrane, and they are hydrophobic. So what happens is that these naturally form a lipid bilayer in an aqueous environment, and that's because these inner pieces that are hydrophobic can protect themselves from water, and these outer pieces can be in contact with water. So sometimes you see phosphate diagrammed in the form where you have a circle at the head and then two fatty acid tails. And so if you were to overlay that on the chemical structure that you see, this head group right here consists of this head group and this phosphoryl head group right here, and then the fatty acid tails down at the bottom. So we get to go into a little bit more detail about the structure of the fatty acids now, and membrane lipids. A couple of things that you should know about. To synthesize the fatty acids that form the lipid bilayer, you start out with you start out with glycerol that contains these hydroxyl groups. And then you start out with also a fatty acid, which contains a carboxyl group at the end. And when the glycerol and the fatty acid interact, it forms an ester linkage. So what exactly is an ester linkage? An ester linkage is a bond between a hydroxyl that is an OH group and a carboxyl group. That is a COOH. And it takes the general form that looks like this. So here you have a carbon double bonded to an oxygen, and then you have an R group, single bond to an oxygen, and then another R group. So you can see how this general structure corresponds to the structure that you get when you link a fatty acid with the glycerol. Now, recall that our membrane has not only it has a head group and it has two fatty acid tails. So if we were to draw those on here, the head group would be coming off of this carbon. And if we were to draw the other fatty acid chain, it would be right there. So here is another picture of a membrane, and the reason that I show this to you is because the way that they've drawn this picture kind of gives you an indication of the fluidic nature of membranes. So membranes are actually really quite fluid. The proteins actually float in this two-dimensional sea of phospholipids. So fluidity is required. You can't have a membrane that is rigid and that the proteins can't, move around in. So the membrane fluidity is affected by the lipid composition. And in the next several slides, we're going to be talking about how lipid composition and other alterations to the membrane can impact fluidity. So first of all, let's say that an organism wants to increase their membrane fluidity. They want their membrane to be a little bit more fluid. There are a couple of ways that organisms do this. And one way is to use lipid molecules with shorter chain lengths. And let me just draw that out. So here you have a diagram of a lipid molecule with shorter chain lengths. And here is a diagram of one with longer chains. And it's this one that makes the membrane more fluid. And the reason for that is because in these fatty acid side chains, there are van der Waals interactions. And in the case of the shorter chain lengths, there are fewer van der Waals interactions, which increases the fluidity, makes it more fluid. Okay, So the second way that membranes can increase their fluidity is to use more double bonds, and that is unsaturation in the lipid chain, and that creates kinks. So let me show you how that works. Let's just take an example of single bonds. We'll start out with single bonds first of all. So here's single bonds between the carbons. And these single bonds, these are saturated with hydrogens. So you've heard the term saturated fats and unsaturated fats. So saturated fats are fats with single bonds, and so they've got a lot of hydrogens. So I'm drawing them in right there. Now, let's say that we have a double bond, and the double bond adds kinks. And let's draw that. So here you have, let's draw the example here. We come to a carbon, and then you have a double bond right there. And in the case of cis double bonds, it looks like that. So there's a cis double bond, and there are fewer hydrogens here because this carbon has 1, 2, 3 bonds, and there's only room for one more. Assuming that this is a single bond, there's room for one more hydrogen instead of 2. So it's more unsaturated, there are fewer hydrogens. And this kink makes it so that the lipid molecules can't pack in tightly together, and therefore it increases the membrane fluidity. Make that a little smaller because I'm kind of running out of space. Now I just want to give you an example of another type of double bond, because I don't want you to confuse cis and trans double bonds. It's the cis double bonds that add kinks, and the trans double bonds do not add kinks. So another way that double bonds can occur, we'll start out the same way. And trans just continues along its merry way. Trans, no kinks. So it's through the cis double bonds that membrane fluidity is increased. Both are unsaturated, both cis and trans are unsaturated, but it's really the kinks that increase the fluidity. Now, let's say that a cell wants to decrease the membrane fluidity. That is, it wants its membrane to be a little bit more rigid. Well, there are several ways that you can do that. And of course, the first one is use lipids with longer chains. So for the exact same reason that shorter chains increase membrane fluidity, longer chains decrease membrane fluidity. And again, you can use, to decrease the membrane fluidity, you can use molecules with fewer double bonds. So that is with fewer kinks, and that means that everything can pack tightly together. and that decreases the membrane fluidity. It makes it more rigid. Now, there are a couple of other ways that you can decrease the membrane fluidity, and that is you can add reinforcing agents. So you can add molecules to the membrane that make it more rigid. Now, you may have heard of sterols, and an example of a sterol is cholesterol. So in eukaryotes, sterols perform that function of making the membrane more rigid. In prokaryotic organisms, hopanoids are used. So the arrows aren't matching up exactly like they should. But here are the hopanoids, and these are the molecules that hinder movement of phospholipids in prokaryotes. And just for an example, this is cholesterol, which makes eukaryotic membranes more rigid. And to drive the concepts home, we're going to talk a little bit about some fats that you may be familiar with. These are fatty acids, and the same principles apply to, say, fats that you cook with and fats that you eat, to the lipid and fatty acids that we're talking about here. So let's first talk a little bit about butter. So butter at room temperature is a solid. And if I were actually in lecture, I'd stand in front of the class and ask you questions and let you be silent until someone finally pops up with an answer. I can't do that here. So what I'd like you to do is, before I give you the answer, at least try and think about the answers to the questions. So my first question is, okay, so butter is a solid. Does that mean the membranes or the fats are more or less fluid? They're less fluid, they're solid, and they fall into this category. So that means that butter is what, unsaturated or saturated? Butter is saturated. And there are very few or no double bonds. Now let's talk about vegetable oil. And at room temperature, vegetable oil is a liquid. So that means that it's more fluid. So in the vegetable oil, are these going to be saturated or unsaturated? It's going to be unsaturated. More double bonds. So I like this little picture at the bottom right-hand corner of the screen, because it shows the difference between what a fluid membrane looks like and a rigid membrane looks like. So here is an example of a fluid membrane, and you can see here it's diagrammed how the cis double bonds make kinks in the in the lipid chain, and makes it so they aren't packed as tightly together. You don't have the van der Waals forces. It's more fluid. However, if you have saturated chains plus hopanoids, then you have a very tightly packed membrane, which increases the solidity or decreases the fluidity. So I've gone through all of this, and the reason it's important is because cells use membranes to adapt to different conditions. There are some cells that live in very hot environments, so even at water temperatures that are above the boiling point, and there are cells that live in very cold environments. So what I want you to think about is, when would a cell need to have increased membrane fluidity? So if you want a highly fluid membrane, So if you let me think about that in a different way. So let's say that you're a cell and your membrane is very fluid. That is, it speaks for itself. It's very fluid. So what would happen if you stuck a cell with a very fluid membrane in a hot springs environment? What happens when you overheat butter or oil? It just wants to fall apart. It isn't going to maintain its integrity. However, if you put that same cell in a very cold environment and it has a very fluid membrane, the cold is, and think about what happens when you put butter in the fridge. It makes it really hard to spread. So if you have a cell that lives in a really cold environment, you're going to want to have a membrane that is very fluid, that still allows that fluidity, allows the proteins to move around. It isn't going to be frozen rock hard. In the other extreme, let's say that you have a cell with a very rigid membrane, and you were to expose the cell to extremely high temperatures. It would be able to keep its membrane integrity in the high temperatures because of its rigidity. So I can guarantee you that these types of questions are going to be on the exam, and you'll need to think about instances when a cell would want to have high fluidity or low fluidity and the circumstances behind it. So what I'd like you to do on your own time is to think about this particular question and see if you can figure out an answer to it to your satisfaction.